Welcome to all of you in today's session where we are going to learn about the final and most significant stage occurring in the inner mitochondrial membrane, that is oxidative phosphorylation. You see that cellular respiration is a vital process in which cells basically extract energy from uh, different nutrients to produce adenosine triphosphate, which serves as a primary source of energy in the cell. And basically, it generally involves a series of complex series of biochemical reactions that takes place in the three main stages, namely glycolysis, the citric acid cycle or Krebs cycle, and oxidative phosphorylation. That includes electron transport chain and chemiosmosis. Now you see that we have gone into details of glycolysis. Basically, this initial stage occurs in the cytoplasm and involves the breakdown of one molecule of glucose into two molecules of pyruvate. Here you see that one molecule of glucose is broken down into two molecules of pyruvate, and generally glycolysis yields a very small amount of ATP and NADH, and then this pyruvate is further utilized in the process of another cycle known as uh, Krebs cycle. We have gone into detail the Krebs cycle or citric acid cycle, but the seat of this Krebs cycle is different from glycolytic pathway. The seat of basically Krebs cycle or citric acid cycle is mitochondria, and the citric acid cycle further oxidizes the pyruvate produced in the glycolysis. So basically this citric acid cycle generates additional NADH and FADS2, and carbon dioxide is basically released as a byproduct. Now third one is oxidative phosphorylation, and this is the most you can say that most significant stage that occurs in the inner mitochondrial membrane. And you see that the seat of all these three processes, namely glycolysis, Krebs cycle and electron transport, is different. And you must remember at this point that this electron transport chain or oxidative phosphorylation takes place in the inner mitochondrial membrane, and generally in the oxidative phosphorylation, electrons generally from NADH and FADS2 are transferred through a chain, and then releasing a sort of energy. So this energy is basically used to pump proton across the mitochondrial membrane and then creating an electrochemical gradient. So you see that the flow of protons back into the mitochondria occurs through ATP synthase, which drives the synthesis of ATP in the process known as chemiosmosis. So overall this cellular respiration is a very highly efficient process, and that extracts energy stored in glucose and other organic molecules, converting it into ATP that is required by the cell. And the complete oxidation of glucose in cellular respiration produces you see that carbon dioxide and water as byproducts. Now the most crucial step we will discuss today is electron transport chain, which is a very critical component of cellular respiration, generating energy in all the living organisms. Generally, as I mentioned earlier, that electron transport chain takes place in the inner mitochondrial membrane of eukaryotic cells and plasma membrane of prokaryotes, and plays a crucial role in converting energy stored in various molecules into a form that could be utilized by the cells, namely ATP. And generally in electron transport chain generally occurs through a series of redox reactions involving electron carriers, and in this way this harness energy and then release by transferring electrons along a molecular pathway and then ultimately driving the synthesis of ATP that serves as a primary currency of the cell, energy currency of the cell. So this intricate process is very much fundamental for various cellular activities, you might be aware, for carrying out the basic metabolic functions to complex physiological processes. Now, electron transport is basically serves as a final stage of cellular respiration, and as I mentioned earlier, that it occurs in the inner mitochondrial membrane, and then you see that we are going to discuss in detail the molecular electron transport and energy conversion in this whole process. And basically you might be aware that ATP serves as energy carrier. So ATP is indeed a very crucial molecule in the cell, serving as the primary carrier of energy. It is stored and releases energy during cellular respiration. Now you see that in citric acid cycle or Krebs cycle is generally a series of chemical reactions takes place in the mitochondria, where generally acetyl coenzyme A derived from the breakdown of carbohydrate, fats or protein is basically oxidized to produce carbon dioxide, NADH, FADH2 and ATP precursors like GTP. And this cycle you see that is a very key part of cellular respiration. Now in Krebs cycle generally NADH and FADH2 formation also takes place, and you see that during citric acid cycle generally these NADH and FADH2 are produced as high energy electron carriers, and these molecules then later participate in the process of oxidative phosphorylation. Now question arises, what is oxidative phosphorylation? So this is an oxygen dependent process that takes place in the inner mitochondrial membrane as I mentioned earlier. So basically it involves the transfer of electrons from NADH and FADH2 through a series of protein complexes known as electron transport chain, and generally the energy is released during this electron transfer is basically used to pump protons across the inner mitochondrial membrane and creating a proton gradient. Now how ATP is synthesized? So the proton gradient created by electron transport chain is basically used to drive synthesis of ATP through a protein complex called ATP synthase. And basically this process you see that is known as oxidative phosphorylation, because it involves the phosphorylation of ADP to form ATP, and generally this process occurs in the presence of oxygen. So now a basic idea should come in your mind, what is oxidative phosphorylation, how it is different from photophosphorylation and substrate level phosphorylation, because all these two aspects I have discussed earlier while discussing the glycolysis and photosynthesis. Now the electron transport chain, as you see that occurs in the inner mitochondrial membrane, and you see that mitochondria consist of two membrane, outer membrane and inner membrane. The outer membrane and the space between outer and inner membrane is called intermembrane space, this space, while the space basically enclosed by inner mitochondrial membrane is referred to as mitochondrial matrix. So the inner mitochondrial membrane is the location where protein complexes are of electron transport chain are basically embedded. You see these are the different complexes that are embedded in the inner mitochondrial membrane. You see that these complexes basically include NADH dehydrogenase complex 1, you see NADH dehydrogenase complex 1, then succinate coenzyme Q, reductase complex complex second, and then cytochrome BC1 complex third, and then cytochrome C oxidase, this one complex 4, and ATP synthase complex 5. So each of these basically complexes plays a very, you say, crucial role in the transfer of electrons and pumping of protons across the inner mitochondrial membrane. So you see that how these complexes, different complexes, these are embedded in the inner mitochondrial membrane, that helps in transfer of electrons from one carrier to another carrier, and accompanied by outward pumping of protons. You see, this arrow shows outward pumping of protons. So you see that this complex 1, 3rd and 4th, they pump generally proton outward, whereas one complex pump proton inward, where one complex second is unable to pump proton, it helps only in the transfer of electron. So these are the very, you see that crucial point you must note at this juncture, that which complex is helping to transfer electron as well as pumping up proton, and which one is only solely responsible for transferring electron. So in this respect, this complex 1st, 3rd and 4th, they pump outward protons, whereas this one complex 2nd is only transferring electrons, not involved in pumping protons. And this ATP synthase is basically very crucial, which pumped proton from inner mitochondrial space into the mitochondrial matrix and drives the synthesis of ATP. I will discuss in detail. Now, how these complexes, they are basically organized for the electron transport chain. So you see that electron transport chain, as I mentioned, that is composed of a series of protein complexes and small organic molecules that work together to transfer electrons and pump proton across the mitochondrial inner membrane. So basically you see that complex 1st is also known as NADH dehydrogenase or NADH ubiquinone oxidoreductase. Basically this complex 1 initiates the chain reaction by oxidizing NADH. Next is complex 2nd, known as succinate dehydrogenase or succinate ubiquinone oxidoreductase, which contributes to the electron flow by oxidizing succinate. And complex 3rd, known as cytochrome BC1 complex or ubiquinone cytochrome C oxidoreductase. Then this complex 3rd further drives the electron transfer, while complex 4, known as cytochrome oxidase or cytochrome C oxygen oxidoreductase, culminates the process by reducing oxygen to water, and this enables basically to the final step in the synthesis of ATP. And together you see that these complexes form a very intricately choreographed system essential for the efficient production of cellular energy. And basically the flow of electrons through these protein complexes is basically accompanied by the pumping of protons across the inner mitochondrial membrane, and this establishes basically a very crucial proton gradient, and then energy is released during the electron transfer is used basically to synthesize ATP through ATP synthase, and this process is known as oxidative phosphorylation. So this the overall coordination and efficiency of the electron transport chain basically contributes very much significantly for the production of cellular energy. Now, what are the basic components or carriers involved in the electron transport chain? So in this regard, there are 5 main carriers involved in the electron transport chain, namely NADH, flavin mononucleotide, coenzyme Q, cytochrome C and cytochrome C oxidase. So, I will discuss one by one what are the important role played by each protein carriers, each carriers involved in the electron transport chain. First one is you see that in the slide is NADH, that is nicotine amide adenine dinucleotide, and this NADH is basically a coenzyme, you might be aware, that serves as electron carrier, and basically this complex 1 NADH carries very high energy electrons from glycolysis and citric acid cycle to electron transport chain, and then in electron transport chain NADH basically donates its electron to complex 1. Then second one is flavin mononucleotide, this one flavin mononucleotide, and this FMN is or flavin mononucleotide is a prosthetic group and is a part of complex 1, that is NADH dehydrogenase, and basically this FMN accepts electron from NADH and transfer them to the next carrier in the electron transport chain. And very important and interesting fact I would like to share with you that FMN is a mobile carrier that moves within the complex to facilitate electron transfer. Now, third carrier is coenzyme Q, that is also known as ubiquinone. This coenzyme Q, also known as ubiquinone, is a lipid soluble molecule, and it is also one of the mobile carrier that transfer electrons from complex 1 and complex 2 to complex 3rd. And this coenzyme Q is the only electron carrier in electron transport chain that is not protein bound prosthetic group. It is a carrier of generally hydrogen atom that is protons plus electrons. So, you must note this very differentiating point. Then cytochrome C, this cytochrome C is a very small heme containing protein and located in the intermembrane space of the mitochondria, and it accepts inner membrane space of mitochondria, and it accepts electron from complex 3rd and transfer them to complex 4. And this cytochrome C is basically a water soluble carrier that moves freely in the intermediate space. This is also very interesting to note here. And then cytochrome 5th is cytochrome C oxidase or complex 4. You see that this complex is the final complex in the electron transport chain, and basically this receives electron from cytochrome C and transfer them to molecular oxygen and then reducing it to water. So, basically the transfer of electrons through complex 4 is coupled with the pumping of proton across the inner mitochondrial membrane, contributing to proton gradient, which is used to drive the synthesis of ATP. So, in nutshell, these 5 carriers, namely NADH, FMN, coenzyme Q, cytochrome C and cytochrome C oxidase complex 4, these work together to transfer electrons through electron transport chain, generating a proton gradient across the inner mitochondrial membrane that drive the synthesis of ATP. Now, I will specifically discuss how these different complexes they are localized in case of plants. So, basically in plants there are additional NADPH dehydrogenases, both external and internal they are present, which oxidizes NADPH by transferring electrons directly to ubiquinone Q, thus missing the first step of proton translocation. First step of proton translocation, you see here. Generally UKS2 is oxidized by complex 3rd, which is basically coupled with translocation of 4 protons across the membrane, In plants, there is also an alternate oxidase, you see, alternate oxidase present, which can oxidize UQS2 directly without transferring electrons to complex third and complex 4. Then from complex third, generally you see that electrons are carried to cytochrome C and are delivered to complex 4, which pass on the electrons to oxygen, which is reduced to water after receiving electrons from combined with protons from the mitochondrial matrix. Now, it is very interesting to share that how this complex 1, that is NADH dehydrogenase, is localized, how it is made up of. So, I will discuss the key feature of complex 1. First is basically, as we talk about the structure of the complex 1, that is NADH dehydrogenase, also known as NADH ubiquinone oxidoreductase, consists of about 45 polypeptides. Basically, this complex consists of how many peptides? 45 polypeptides, and you see that shape of this complex 1 appears more or less L-shaped, and this resembles to mammalian and fungal complex 1, but it has certain plant specific proteins. So, this mitochondrial generally genome encode this complex 1, and the generally more than 9 components of complex 1 are encoded by mitochondrial genome. Then cofactors, basically it contains FMN containing proteins, and at least 6 iron sulfur centers are also present in this complex 1. Now, how this complex functions in the oxidation of NADH. So, complex 1 basically oxidizes NADH, which is produced in the mitochondrial matrix during the tricarboxylic acid cycle, and then basically electrons are transferred to ubiquinone. You see, the electrons are transferred to ubiquinone. So, electrons basically that are removed from NADH are transferred to ubiquinone, also known as coenzyme Q, and the transfer involves a sort of conformational change in the antiporter proteins present in the membrane of the complex. So, then how proton translocation occurs? Next question through this complex. As you see that electrons are transferred to ubiquinone, as I mentioned here, electrons are transferred to ubiquinone. This leads to conformational change in the antiporter protein, leading to the translocation of basically 4 protons across the inner mitochondrial membrane. You see, 4 protons are translocated from the mitochondrial matrix to the intermembrane space, and this proton translocation is generally coupled with the electron transfer and contribute to the establishment of a very crucial proton gradient. Now, this complex basically catalyzes the transfer of also one hydride ion from NADH and one proton from the matrix to ubiquinone, and then this complex also help in coupling with proton pumping. Generally, the transport of 2 electrons to ubiquinone by complex 1 is coupled with the transport of 4 protons across the membrane. So, this complex basically serves as proton pump, contributing to creation of electrochemical gradient, which is used for the synthesis of ATP. So, in nutshell, it could be summarized that complex 1 plays a very crucial role in the mitochondrial electron transport chain by oxidizing NADH and transferring electrons to ubiquinone and contributing to the proton gradient across the inner mitochondrial membrane. Now, complex 2 is also known as succinate dehydrogenase or succinate ubiquinone oxidoreductase, which is a membrane bound enzyme in the tricarboxylic acid cycle, responsible for catalyzing the oxidation of succinate to fumarate. And this complex also plays a very crucial role in electron transport, and this complex second is distinct from complex 1 and is very integral to the electron transport chain. And I will discuss how this complex 1 impart different functions like catalytic functions. This enzyme basically facilitates the oxidation of succinate and extract 2 electrons and then transferring them into ubiquinone and resulting in the reduction of ubiquinone to ubiquinol or UQS2. Whereas you see that complex second does not contribute to proton translocation across the mitochondrial membrane. So, in this aspect, this port complex second does not contribute to the creation of proton gradient, as it only involved in the transfer of electrons, not outward pumping of protons. So, in this aspect, this complex second is different from complex 1. You should note all very differentiating point. So, complex second is the smallest complex within the electron translocation, or you can electron transport chain, comprising only 4 different protein subunits, and these the size of these subunits ranges from 13 to 70 kilodalton. Generally, in majority of the plants, including angiosperms and gymnosperms, none of these proteins are encoded by mitochondrial genome. So, this is very interesting point to note at this juncture. And how these proteins are arranged, basically it consists of 2 smaller hydrophobic integral membrane protein and 2 larger hydrophilic proteins. Then this protein further extended in the mitochondrial matrix. Basically this succinate binding site, that is flavin adenine dinucleotide, and then 3 iron sulfur centers are associated with the peripheral proteins extending toward the mitochondrial matrix. Additionally, the hydrophobic integral membrane protein generally have 2 binding sites for ubiquinone, this one ubiquinone, along with the association with cytochrome B. Then how this complex is helpful for electron transfer? So, basically this complex second generally extract electrons from succinate and then transfer to ubiquinone through FAD and 3 iron sulfur centers. This role of basically cytochrome B is not in the main pathway of electron transport, but it is believed to prevent the leakage of electrons to oxygen, thereby helps in mitigating the formation of reactive oxygen species. So, this is all about complex 1 and complex 2 involved in the electron transport chain. I will discuss further journey of electron transport and proton pumping and other complexes involved in the electron transport chain, and then finally I will discuss also respiratory quotient in my next lecture. Thank you very much.